as I told you in the last second year class, second prof examination, there is a full length question paper on what I have discussed yesterday and what I will discuss today also, part of it. I am going slowly because you understand the matter. And wherever you cannot understand, I please note down again, I am telling you, and ask me, interrupt me at any point of time, and or SGD, I will take one SGD, at that time you can discuss, or anywhere I am moving in the campus or in the class also, practical class, you can ask, taking that note that, sir, I could not understand that thing. I will try to explain you my level best. So, human anatomy part, as you know, which part of the body has been covered in anatomy so far? Which one? Upper limb. Upper limb only. Thorax is complete. Abdomen? No abdomen. So, okay. So, we are having the basically this innate system. We are having the basically originally the T cells and B cells are there produced from the bone marrow, and the T cells get processed at the thymus, and they are also localized in the Pearce patch, and these are the cells which I have said on that first day. So, that contains the innate immune system, and some of the cells which are produced from the bone marrow, they are processed to the lymphatic system, the lymph nodes. They reside on the secondary organ. So, bone marrow is the primary organ where it is synthesized, and these are the secondary lymphoid organs. And the secondary lymphoid organs here, B lymphocyte and T lymphocytes are there. So, T lymphocytes originate basically from thymus. And our body, in our body, we are having lymph nodes. You have noted, you have seen in your body, you will be able to find out. The cervical nodes, you can see all the occipital node. I can, or you can palpate my occipital node at any point of time. I am having one occipital node, lymph node. So, lymph nodes are present, and they are actually the organ where the actually infection is controlled. So, this is the anatomy of the immune system. So, write down host defenses. This is local or systemic. Local I will tell in detail. Systemic means what you will get, all of you are immunized, you got a systemic defense. It can be a specific one and it can be a non-specific one. And there are two main types, one is humoral type, one is cell-mediated. Humoral is antibody-mediated and cellular is cell-mediated, that is T-cell mediated. Now, actually there is no isolated absolute isolated compartment of the two systems. Humoral absolutely no, humoral they require some help from the T cells. Similarly, cellular immunity, that is not absolutely cellular, some amount of humoral component is also there. So, there is overlapping host defense mechanism. They operate either alone or in combination depending on the situation. And what is hybrid immunity? Today you know one is going on online, one is going on offline. So, when anything is held, any conference is held both in the online and offline, we call this hybrid mode. Similarly, hybrid immunity, what is hybrid immunity? Something you have acquired from the nature or from the prior infection, and same thing has been vaccinated to you. So, you get a dual immunity, that is called hybrid immunity. That is natural immunity which you have developed, and you have been given artificial immunity also. So, immunization, that is called hybrid immunity, that is terminology. So, immune response basically, as I told you, the bone marrow is the products where the hematopoietic stem cell is there. I will tell you from how these lymphocytes are developed. So, write down from bone marrow, lymphocytes are produced, and they are produced at the thymus gland, and they produce T lymphocytes. and T lymphocytes can directly kill the target cells, and they are called effector cells of the cell-mediated immunity. So, killing is basically affecting the mechanism. So, they are basically killer cell or effector cell. Don't get confused with the natural killer cell, which are present in the immune system, innate immune system. So, in the peripheral blood, you just note down T lymphocytes are 80% and B lymphocytes is 20%. কি হলো, কিছু অসুবিধা হচ্ছে? হ্যাঁ, বলছি আমি এক এক করে এক্সপ্লেন করছি। See, where from this B term has come, T has come from thymus, no doubt. But this B term has come from the bursts of fabrices. Actually, most of the research work have been done on the animals, experimental animals, rats, rabbits, other animals also. And then from there we learn something and we try to find out similar things in our human body. So, these are basically present in the bursts of fabrices. This B cell is coming from bursa of fabrices in avians. So, they are having in the proctodial region there is a dorsal live-reticulum. So, that is their site of hematopoiesis. Now, ours is in bone marrow, they are having also bone marrow. So, these cells are processed there. So, there is a plication of luminal surfaces just like our intestinal system also. So, this is chicken. So, the Barsa equivalent organ we are having Barsa equivalent organ in our system, that is called in the gut. You are having Pierce patches in the anatomy, you will read it. Pierce patches I will discuss so that you understand, correlate also while you study anatomy. And the lung associated lymphoid organs. I will tell in detail about the mucosal lung associated lymphoid organs or the gut associated are basically related to mucosa. And there are a number of mucosa right from our nostril, mouth till genitalia and GI system. So, B cells do not produce plasma cells, immunoglobulins. They are converted into plasma cells as I told you earlier. So, that plasma cells form immunoglobulin and this B cell governs the humoral immunity. Hey, just look at this thing. See, we are having a human beings. This is a mouse model is always taken. You will see many experiments are with mouse model. So, mouse we are in this is the intestine. If you look at this structure, they will get small amount, but number is huge here. So, these are called Pierce patches. Say this is the intestinal epithelium. You have seen some slides in the histology. Histology you have seen. So, this is on the surface you will say the epithelium, this is a schematic diagram, not exactly histological diagram. So, just to say you that this is the actual Pierce patch, schematic, absolute schematic diagram. So, these are the cells whatever I have said in the innate immune system, they are all present. So, this is a mucosa associated lymphoid tissue, its name is MALT. So, gut associated lymphoid tissue is called GALT. They are basically Pierce patches. So, as I told you in the first slide, there you remember that we have said that we are having the externally skin and the interiorly mucous membrane, epithelium. So, this mucosa associated lymphoid tissue is called MALT, and you are having a number gut associated, bronchus associated, nasal associated, conjunctive associated, larynx, skin, vulvovaginal, testis, and also have you been told about tonsil and upper part head neck has been covered? No, you don't know that then that thing. So, when you will be told about this oral cavity, you will see we are having two tonsils on either side. So, similarly we are having one tonsil at the nasopharynx, that is called adenoids, and then when the eustachian tube through which the air is communicating with the nose with the air, if you blow it, if you close your nose and if you blow it, you will see that air is hitting the tympanic membrane. So, where it is opening in the nasopharynx, we are having a tubal tonsil, and then lingual posterior surface. If you protrude your tongue too much, you will see some of the structures at the back of the tongue, round shaped structure. These are all lymphoid organs. So that produces Walder's tonsillar ring. The tonsillar ring, there is a ring formation. On the top you will get that adenoids, and then tubal tonsils, then palatine tonsils, what you know, and then lingual tonsil, and they form a circle. So they act as a protective, first line sometimes defense, or something is entering into our own system, either through the nasopharynx or through the gut. So they are basically regulate local mucosa. They are protecting, regulating mucosal immunity. Because otherwise, if that is invaded, then a infection will occur. So clinical significance, why I have written basically, because this is a site of lymphoma, cancer cell. So where if there is any tumor in these cells, you think of lymphoma, and this is a non-Hodgkin lymphoma. It is not a good thing, good cancer at all. This cancer is basically bad, so this is a site for lymphoma. Where lymphoid tissue are here. So if the lymphoid tissue undergo malignant transformation, that is called lymphoma. Write down one thing, that is mucosal associated lymphoid tissue, and then you remember whatever you can say. So primary lymphoid organs, note down this part. So primary lymphoid organ is basically bone marrow and thymus. From bone marrow, hematopoietic stem cell is there. So they are producing B cells, and once the B cell is formed, B cell is thrown into the circulation, and T cells are also processed at the thymus. So the difference between the two, this melt, that is mucosal associated lymphoid tissue, they are uncapsulated. They don't have any capsule. They are just, if you look at the structure, there is no capsule. But in contrast, this lymph node, they are having a capsule around it. So this is uncapsulated, this is capsulated. So secondary lymphoid organs are basically lymph node, spleen and melt. So lymph node and spleen is encapsulated, spleen is a structure which is encapsulated. So immune response occurs in these secondary lymphoid organs. Now you note down, this will be recorded on the physiology. Physiology class have been told about this, then it is difficult really. That is the reason I am going very slow. See, from the bone marrow, we are having pluripotent stem cell. That is hematopoietic stem cell, totipotent cell, pluripotent cells, multipotent cells. There are many types of cells that we will discuss. So from pluripotent cells, we will get two types of main things. One is common lymphoid progenitor. Write down this thing, it will be helpful. Lymphoid progenitor, and one is common myeloid progenitor. So from the lymphoid progenitor, we get cells, T cells, B cells, and they are involved in the adaptive immune response. And in the myeloid series, we get all rest of the cells, the neutrophil, eosinophil, basophil, that you know. In addition, note down immature dendritic cell, then mast cell, precursor, monocytes you know, then platelets and erythrocyte. So basically lymphocyte, T lymphocyte and B lymphocyte, they are produced from the lymphoid progenitor cells, and both are involved in the adaptive immunity. The rest of the cells are involved in the innate immune system. So from that progenitor cells, basically they are finally processed into lymph nodes. This adaptive immunity is a lymph node, and they are present in the tissues, many tissues, all the tissues. So in the blood we are having neutrophil, eosinophil, basophil, monocyte and lymphocyte. Only lymphocyte is there in the lymphoid organ. Rest myeloid series, and in addition there is a dendritic cell, immature, and mast cell. So, these are the all the cells are present in the circulation. But in the tissue, if you get, ultimately you will get this tissue in the lymph node, and in the other tissues you will get all these type of cells. And the immature dendritic cell gets mature in the lymph node, and they are antigen present in the cells, just like monocyte. Antigens are not presented immediately, as you will understand in a little while. I am telling you because immature dendritic cell will get into mature dendritic cell into the lymph nodes. Now, there are immune cell types, as I told you in the beginning: macrophage, mast cell, dendritic cell, neutrophil, eosinophil, basophil. Only note down a part of the function, not all. So, one in phagocytosis. You note down, not everything you are required to write down. Phagocytosis, basic, macrophage and neutrophil. They are in, at least you must be able to tell something about this thing, what is the function. Phagocytosis means they are phagocytosis, that you have know very well. So, function of macrophage and neutrophil is predominantly phagocytosis. In addition, they are having other functions. At least you remember, histamine production is related to allergy. We itch when we get something, and that is due to histamine production, and or bronchial asthma. That day I was coughing, maybe today I will be also coughing. I have brought many things. So, from our mast cell, histamine is produced, and mast cell or eosinophil, they produce histamine. So, when you are having allergy, as I told you earlier, the eosinophil count is increased. Eosinophil count is increased because of the allergy, not vice versa. Eosinophil count has increased, that is why you are having allergy. No. So, cytokine production, you just write down mast cell, dendritic cell, neutrophil, eosinophil and basophil. See, apart from this, there are many other things. I do not want to bother you with too much information. Neutrophil extracellular trap is another mechanism for destroying organisms. I will not tell this thing. Regina, एक बार प्रेस करो, ओखान तक प्रेस, आई देखो. Each cell, as I told you earlier in the class, that each cell is having B cells. B cells secrete immunoglobulin. B cell secretes immunoglobulin, and one part of that same immunoglobulin is present on its surface. That is B cell. That acts as a receptor. It will bind to the antigen. Similarly, T cells are having T cell receptors. Write down highly specific cell surface receptor, because if they cannot specifically bind antigen, the immune system will not be operative. So, T cell receptors are called TCRs and B cell receptors are called BCRs, and they actually recognize whether it is a self molecule or non-self. If it is a non-self, if it is dangerous, they will mount an immune response. If it is not, it will be innocuous, means it is not dangerous, then no issue. So, they recognize specifically, you write down whatever you can try to remember, you remember. So, T cell receptors, they are having, T cell is having a, as I told you, it can kill directly and it helps in antibody production. Remember, here you note down only one thing, a clone. Clone you write down, clone is identical cell derived from a single progenitor. This part you write down. What do you mean by clone? Rest you try to understand. Most important part is clonal selection and expansion. I will spend at least 5 minutes time on this. Clone is an identical cell derived from the same cell, just like a photocopy. From the same copy, you are getting multiple, from the same document, you are getting multiple copies. That is what is clone. Each B cell is programmed to produce only one type of antibody, not more than one, because it is having an unique receptor on its surface and it will produce the same antibody. And in the B cell, and the first class itself, I told it is having a nearly 1 lakh receptors. That means the picture what you have seen, this is each B cell is having a 1 lakh receptor 10 to the power 5. So what happens basically after antigen binding? Now look at this picture, you remember this one. See, this is the single B cell. Suppose there is an antigen and it got activated antibody receptor, it has interacted. Now from this cell, 2 cells will be produced. From these 2 cells, another 2 cells. From these 2 cells, another 2 cells. From this cell, another 2 cells. This is clonal expansion. This is clonal expansion. From a single cell, you are having a number of cells. Some of the cells, if it is a B cell, suppose some of the cells will form antibody, and rest will be there in the as memory cells. From here I can show you this structure I am telling you. Suppose here we are having 4 cells. Suppose this here. So as I told you, some are producing antibodies, some are producing memory cell. Maybe these 2 cells are producing antibody. These 2 cells may be memory cells, or these may be producing antibody, but this is a memory cell. This is a memory cell, this is producing antibody. It can so happen. So at the end of clonal expansion, we get memory cells and the antibody producing cells. So the antibody secretion will be much higher because the number of cells has increased. Now this is done in the primary immune response. So in the primary immune response, we get a memory cell, this antibody, we get a small quantity of antibody, and the number which was present only 1, the number will increase to many-fold, many times, and they will be stored in our body as memory cells. Now next time when you give the antigen, so there will be multiple cells and the antibody titer will be very high, depending on the number of memory cells. Do you get my point? So in the primary response, the antibody concentration in case of antibody, it is called titer, not concentration. So in primary response, antibody titer will be low because of the numbers are low. Because at this stage, this much many numbers are formed. Now when the secondary response is there, So numbers will be 5 because this number of which is 1, it may be of the order of 1 lakh or 10 lakhs or 1 million, 10 millions. So 10 millions will be start producing antibody immediately. And here you will get some amount of time, 4 to 5 days, even 6 to 7 days, 10 days, 2 weeks, 3 weeks time you will get to get this thing. So some time will be required here. Here that time will not be required. Here it will start immediate action and then go on proliferating. So then, in this primary response, the lag period means the time from where you got the infection and you get the antibody. That is the lag period. So lag period will be longer here, lag period will be very short, and it will be a sluggish reaction, but it will be very prompt one. I will tell you in the difference. So do you get my point? So this is basically B cell. Similarly for T cells, T cell is having the T cell receptor. So again this T cell, if it is a cell dependent cellular immunity, so T cell will encounter and they are having a receptor but not this antibody. So that receptor will undergo clonal proliferation like this, and at one point of time they will be producing T cells which will be really active. They will be killing the cells, they will be having regulatories, they will be helping, there are many functions which you will know, and some will be stored as memory cells. So mechanism will be the same thing. Clonal expansion is the same thing. Only function here it is B cell is producing antibody and T cell is producing T cells functions. That you will understand. Is this part clear? Now so this is clonal expansion. Now just note down this what I have written. See, this is basically we are having in our body this much amount of cells, suppose. But I told you in our system we have only one antibody and a lymphocyte receptor, that it has encountered an antigen. Now this cell has undergone proliferation, the picture which I have shown, just match it. Now from 2 to 4, now these are, here you see 8 number of cells are there. So out of 8 number of cells, you can see these cells are secreting antibody, and these small cells are remaining present as memory cells. Is this part clear? So this is clonal selection. We are selecting that out of so many things. We have selected this one because this cell surface is matching with that corresponding antigen, not the others. We are having, this cell will react with some other things. Is this part clear? So this is clonal selection, number of first part, and then expansion, and then creation of memory cells. So what is basic structure, again I will tell one in the antibody class. For understanding at this stage, you see so it is just like a Y-shaped structure. Each and every point I will tell once more, just at this stage for understanding. So it is having a heavy chain, note down, one is heavy chain, one is light chain. There are 2 heavy chains and there are 2 light chains. Heavy chains because this, as you can see, this structure is bigger, so this is heavy and this is light. So either it is there are 5 different types. One is alpha, then mu, first is mu, alpha, gamma, delta, and epsilon. This I will tell. Don't be hasty. Right now you understand only this antibody part. So this whole antibody doesn't get involved in the binding part. Only this part, that is called FAB region, means fragment which is binding to antibody. So, there are two light chains, there are two heavy chains. This light chain will be either kappa type or lambda type, not both, mixture. Remember, the light chains will be, heavy chains are of 5 types. I will tell once again, light chains are of 2 types, kappa or lambda, but in a given antibody molecule, either kappa or either lambda, not others. And they are present in this part, and this part is the antibody binding site. Rest part is having other functions. So, you can see here, can you see? Just correlate. The antigen binding site is this one. So, there are 5 types, as I told you. This is a primary basic structure. Now, this IgA molecule is a dimer, means 2 molecules have been joined together with a J chain. Similarly, IgM is a pentamer. There are 5 same structure bound with a chain. So, you are having 2 binding sites here. Here the binding sites are 4, here binding sites are 10. Get my point? So, the affinity, this avidity, the antibody will bind here, antibody will bind here. So, many antibodies and antigens can be bound to that IgM molecule. So, there are 2 terms, note down this one, write down. One is affinity and one is avidity. Avidity means strength of binding between an antigen and an antibody on a single site. Single site. Only one site, single binding site, it is strength of attraction, basically strength of binding or intrinsic affinity it is called. Avidity is basically binding between 2 molecules with the valency of interaction and the total number of binding sites. As you can see that IgG is having 2, IgA 4 and pentamer is having 10. So, affinity, if it is a IgG, IgD and IgE, they are having only 2 binding sites. So, they are having equal intrinsic affinity and intrinsic equal affinity. But in case of IgM, the avidity will be much more. And in the primary response, what happens? Well, less number of binding sites are there. So, avidity will be less. In secondary response, when we get a second infection, since the numbers are very high, avidity will be more. Do you get my point? So, humoral immunity, as I showed, again I am showing this same slide, so that you remember repeatedly. Now, primary response, you just draw this graph. So, primary response, here the time course basically. Basic purpose is to form an immunological memory of primary response. Vaccination or immunity. So, after infection, there is rapid antibody production. You just write this part, these 4 lines, and rest you understand. Formation of immunological memory after reinfection, rapid increase in antibody production, affinity changing and class switching. Affinity changing means avidity now. Initially, we get an IgM and that gets converted into IgG, or it can be IgA, it can be IgD, it can be IgE. So, these are 5 different classes as I told you. So, that class switching occurs during secondary response, but without changing specificity. So what happens, you see here. In the primary response, we get an antibody titer first increased. Now the antibody levels are decreasing. Why? Because every biological material is having a half-life. Okay? So once you have produced antibody, means it will not be present lifelong. Some cells will produce, but the antibody, that protein molecule, will not last for a long time. There is a variable part, and there are lifespan for IgM, IgG, IgD, these are different. So now here you see at this part you are having so many memory cells. Now you got an infection, you get a rapid response of antibody titer. Okay. So the primary response, you get a low antibody affinity antibody, and you get a high affinity antibody in the secondary response. So that just a difference you note down. The entire class could have been covered in 3 classifications only. But now you understand better what I have said so long. So these are points you note down. So primary and secondary, immune response, primary first antigen exposure. Secondary immune response, second subsequent antigen exposure. You all have understood. Now you have to write down these points. I hope you will be able to understand. Antigen processing, I will tell you, it can be both T dependent or T independent. Write down at this stage. In a short while you will know. Because all things are not required. As I told you, there is overlapping. They can operate either alone or in combination. So some cases T cells help will be required, some will not be required. But secondary response T cell is a must. Without T cell you cannot. T helper cells are required. Without T helper cell, antibody cannot be produced. Lag period. As I told you, lag period from the time of introduction of infection to development of antibody. So lag period is longer. Do you agree? Lag period will be longer. Lag period will be shorter. So this is the most important point, is antibody is IgM type in the primary response, and antigen is mainly IgG or other types, other 4 types it can happen. So there is a class switching from one to another, IgM to IgG, other 4 types, mainly IgG, because IgG concentration is highest. Antibody titer, low, do you agree? Antibody titer? Antibody titer will be low or high? Is the primary response? Primary response cells are less, so antibody production will be less. Secondary response, antibody cells are many, so it will be high. You memorize this thing, all these points, you note down during examination. Avidity, I explained, it will be less avid because less number. It is having secondary response, it will be more avid, it is having more numbers. And nature of response, as I told you, it is slow, sluggish and short-lived. And secondary is prompt, powerful and prolonged. নটা 10টা বাজে 5 হলে স্যারকে আবার দেখবে, যদি স্যার আসে তখন আমাকে ডাকবে, আমাকে ততক্ষণ যতক্ষণ পর্যন্ত কভার পারি করে দিচ্ছে। হ্যাঁ, সো ইজ দিস পার্ট ক্লিয়ার? সেকেন্ড পার্ট, this one also you write down. These are 2 differentiating points. What is the difference between active and passive immunization? So far what we have discussed here, this is active immunization. And as I told you in the first class, that if you are employed and if you work for 30 days in a month and at the end of the month you get a salary, you are actively making an income because of your active work. And the same amount of given is by your anybody else, you get it passively. So, passive immunity before birth, we obtain passively from mother. And after birth, you will be doing this thing in the clinical case. Therapeutically, for different kind of diseases, you will be given injection of IgG, immunoglobulin itself directly, or antiserum. In our case, antiserum we use. Now we are not using antiserum at all. For theoretical purposes, I have written. So I am having a violent infection. So maybe tetanus, I am expecting. So anti-tetanus immunoglobulin will be given. Antibiotics will be given definitely to kill the bacteria, but at the same time, so too much antibody will be given to, so that the whatever circulating toxins are there, they can be neutralized. So active outcome of active immune response, it can be infection, nature exposure or artificial. Booster dose, we have not taken any booster dose. For active immunization, all you have taken booster dose. So lag period, passive, there is no lag period because you are directly injecting immunoglobulin. So there is no time. But active some time, duration short lasting. Because as I told you, this antibody is having some amount of life. So here it will be long lasting because the cells will be producing, actively producing. Memory, there will be no memory because you are passively given. So there is no creation of memory cells. You understand what is the actually why I have explained all these things in detail. Clonal expansion, memory cell production. Because now it is easier to understand. Otherwise, this class would have been complete in 15 minutes time by showing these three differences. Now, each and every point I hope you understand. So, memory, there is no memory. Usefulness, immunodeficiency state, you note down. This is clinically important. Because you see, you will be having patients who are having cancers. They do not produce that amount of antibody, or they are not immunocompetent cells. So some cells of the bone marrow, like it can undergo cancer, so they can no longer function. So they are immunodeficiency state. Or some viruses, some bacteria have infected our immune cells. So in that case, we cannot mount any immune response, even if a vaccine is given. Vaccine is contraindicated. Vaccine is not given when the person you know is immunocompromised, and usually it is cancer patients or patients who are taking anti-cancer drugs or immunosuppressive drugs, where we are deliberately suppressing the immunity, or those who are suffering from immunodeficiency states. So here, if we have to give immunity, we have to give passive, because they are unable to produce any immunity, immunoglobulin. So immunoglobulin has to be given. But active, it is not given, and no immunization is given. Remember, in immunodeficiency state, vaccines cannot be given. The role of a doctor at peripheral level is to decide whom to vaccinate, whom not to vaccinate. So they know what are the immunodeficiency conditions, not other stuffs. So you have to decide that this child cannot be vaccinated at this stage. So cell-mediated immunity, as I told you, same thing, that here you note down, in B cells, this part you have understood that it produces antibody. In T cells, as I told you, the functions of the T cells are not same. So there are three different types of cells. One is T helper, note down. T cell secondary response produces T helper mainly. T helper, TC, that is T cytotoxic, and T regulatory. Just wait 1 minute, just wait, and memory cell is there. Here you note down here. So you can see better. So there are 4 different major types. One is helper cell, one is cytotoxic T cell, one is regulatory T cells. You note down only one function. This chart you remember. And memory T cells. Same thing, memory T cells, same memory function. So, helper T cell will help in the antibody production. As I told you, antibody T helper is a must active. So, now you understand helper T cell will produce antibody formation. So, cytotoxic T cell will kill directly. And regulatory cells will be there to regulate. বলো
স্যার টি সেলে তো তিনটে হেল্পার সেল
হ্যাঁ
আর এটা বি সেলে
বি সেলে তো বি সেলের অ্যান্টিবডি প্রোডাকশন করে আর একটা অ্যান্টিবডি প্রোডাকশন করে একটা মেমোরি সেল তৈরি হয় আর টি সেলে দেখো চারটে জিনিস তৈরি হয় একটা তো মেমোরি সেল তৈরি হয়ে গেল ওই বি সেলের মতো আর টি সেলের তিনটে ডিফারেন্ট টাইপ মেন টাইপ এছাড়া প্রচুর টাইপস আছে তোমাদের যদি অতগুলো টাইপ বলে মনে রাখতে পারবে না সেই জন্য সিমিলারলি জাস্ট একটু মিনিমাম নলেজ তিনটে সেল মনে রাখো একটা টি হেল্পার টি সাইটোটক্সিক টি রেগুলেটরি তো হেল্পার টি সেলস আমি আমাদের আবারও বলছি যে আগে বললাম যে বেসিক্যালি টি ইন্ডিপেন্ডেন্ট হয় ইয়েতে কি বলে প্রাইমারি রেসপন্সে বাট টি ডিপেন্ডেন্ট ইজ এ মাস্ট ইন সেকেন্ডারি রেসপন্স সো দিস হেল্পার টি সেলস help with antibody production. Now, if the T helper cells are destroyed, now it will not be able to produce antibody. So, that what happens in AIDS, HIV infection specifically kills helper T cells. So, helper T cells is destroyed means immunity is gone. So, cytotoxic T cells kills viral infected cell. I will show you in a diagram. How do they kill directly? And everything, everywhere, our body, our system, everything is regulated by some means. Even at times you regulate your own activities. So, regulatory activity is there. So, there are some regulatory T cells which does basically the policing action. So, এইখানে লেখো তো একটা cells involved in the cell membrane, directly involved in killing, as I told you, or infectious cells. এই activation of এই তিনটে ফাংশন একটু লিখে নাও। এটা জেনারেল ফাংশন অ্যাক্টিভেশন অফ ফ্যাগোসাইটস করে ফ্যাগোসাইটস দুটো টাইপ জিনিস করে শোনো না একটা হচ্ছে কি ওই ম্যাক্রোফাজ বলবো আর ন্যাচারাল কিলার সেল হুইচ আমি আগেও বললাম এখনো বলছি ফ্যাগোসাইটস দেখো ভালো করে অতগুলো সেল লিখেছিলাম হ্যাঁ আসুন আসুন প্রোডাকশন অফ অ্যান্টিজেন স্পেসিফিক সাইটোটক্সিক টি লিম্ফোসাইটস ইউ ক্যান সি রিলিজ অফ ভেরিয়াস সাইটোকাইন্স অনলি ফাংশনে দেখো এখানে ভালো করে একটা শোনো একটা কথা বলে দিচ্ছি এটা লিখে ছেড়ে দিচ্ছি সো টি সেলস দে আর হ্যাভিং ওয়ান টার্ম ইজ ইউজড নেভ টি সেলস নেভ টি সেলস মিনস ইম্যাচিউর সেলস দে হ্যাভ নট বিন এক্সপোজ টু অ্যান্টিজেন ইয়েট লাইক ইন দিস কেস it is remaining as such, has not encountered any cell, any antigen. So, in case of T cells, that is naive T cell or naive B cell, means it has not come into contact with any antigen yet. Okay? So, with this I am ending, but if I go with this speed, at least 5 classes will not be able to, I will not be able to complete. You can see the slides I can show you. So many slides are there, so many pictures are there, but I am going. Did you understand what I said? So you write down this part. At least this part is written in your book. You understand, and rest of the things I am telling you will not be there. But you understand. Understand as much and write down as much as you can, and you memorize. So with this thing, here I am ending this part. So I think 2 more classes will be required, or in the next class I will finish it off. Chalo, okay. এবার পরদিন পড়া ধরবো বলো 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 কোশ্চেনটা করো কোশ্চেন জোরে বলো এর আগে internal examination. Yes, all what will be told till the examination, this will be all this will be there. You prepare accordingly. And once more I am telling you, if it is not there also, and if you cannot read, again the second class, 2 classes, you will not be able to catch it out. I have spoken very slowly, and only this part has been told, so that clonal expansion you know, is the production of memory cells, and you understand the difference between the two. Now, if I would have said this thing, innate immunity, adaptive immunity, and this two chart, only this, would have been able to understand. তুমি বলো ঠিক করে বলো তো, বুঝতে পারতে? একদম যদি শুধু চারটাই বলে দিতাম, মুখস্থটাই করবে, ওটাই পরীক্ষায় আসবে, কিন্তু ওটা শুধু বুঝতে পারতো, যদি বলে দিতাম, শুধু বলে দিলে হতো কি? এখন তুমি বুঝতে পারছো তো, চারটা ডিফারেন্সটা বুঝতে পারছো তো, কেন কিভাবে হচ্ছে? দিস ইজ দ্য পারপাস অফ কামিং টু দ্য ক্লাস.